Welcome to Hands-On Biology. Watch this lesson. Reproduction. This is the formation of new separately existing individuals of a species from those already in existence. Types of reproduction. There are two main types of reproduction: asexual reproduction, sexual reproduction. Asexual reproduction. This is a type of reproduction in which an individual gives rise to another individual without fusion of gametes. Usually, genetically identical offspring are formed. Vegetative propagation.
Welcome to Hands-On Biology, competency-based learning. Struggling with biology? Not anymore. Here, we break down complex biology concepts into simple, clear, and engaging lessons. Hit that subscribe button now and take your learning to the next level.
Vegetative reproduction or propagation. This involves detaching any other parts of the plant other than the seed, and that part develops into a self-supporting individual. Forms of vegetative reproduction. Natural forms. Artificial forms. Natural vegetative propagation. This is not induced by man, but special perennating organs, which also store food as a means of surviving adverse conditions, are used and develop into offspring. Such perennating organs include bulbs, rhizomes, corms, stem tubes, root tubers, stolon, leaves, suckers, etc. Bulbs. These are modified shoots with a very short stem and fleshy leaves surrounded by scale leaves and with adventitious roots. They contain axillary buds and terminal buds. Each bud produces a new bulb at the end of the growing season. For example, onions. Corm. This is a short vertical underground stem with adventitious roots surrounded by protective scale leaves, e.g. yams. Rhizomes. These are horizontal underground stems, such as couch grass and ginger. Stem tubers. These are underground swollen stem tubers with axillary buds and scale leaves, e.g. Irish potatoes. Runners. They are horizontal surface stems from which adventitious roots and new plants develop at the internode contact with the ground, e.g. in wandering Jew and strawberry. Stolon. Creeping horizontally. Growing stems along the ground surface give rise to new plants at points of stem contact with the ground. Blackcurrant and blackberry. Leaves. Some plants have leaves bearing notches within which buds grow, giving rise to new plants, which later fall off and become independent. Example, bryophyllum. Bulbils. These are aerial tubers that develop from buds in the axil of lateral leaves. When mature, they drop off and grow into new plants. Example in the wild yams. Suckers and offsets. Short horizontal branches originate from the stem just below the ground level. Their terminal buds give rise to new plants, e.g. bananas. Commercial application of vegetative propagation. Due to the increasing population of humans, the natural forms of vegetative propagation cannot produce food to sustain people. Man has improvised means to enhance propagation. These are the artificial forms of vegetative propagation. Artificial vegetative propagation. This is induced by man. Forms include cuttings. The stem is cut into portions, each possessing nodes and internodes, and each portion develops into a new plant when planted. Example in sugar canes and cassava. Grafting. A scion from another plant is fitted onto the stalk of the main different plant of the same species, and the cambium tissues of the two plants are united, making the scion grow into a plant branch with unique combined characteristics, e.g. in citrus fruits. A lemon scion may be grafted on an orange stalk. It can also be bud grafting, where a vegetative plant propagation technique where a single bud, scion, from a desired plant is inserted into the rootstock of another plant to grow as a new shoot. Simple layering. A branch of a tree is bent into the soil, and small slits are cut at the point of contact with the soil. Growth promoters are applied to stimulate root growth. Afterward, a new plant develops from the bent branch. This is common in passion fruits and guavas. Marcotting, air layering, also known as air layering, is a vegetative plant propagation technique where a stem is induced to grow roots while still attached to the parent plant. Once roots develop, the stem is cut and planted as a new independent plant. Tissue culture, or micropropagation, is a plant propagation technique that involves growing plant cells, tissues, or organs in a sterile, nutrient-rich medium under controlled conditions. Key steps: explant selection. A small plant part, e.g. leaf, stem, or meristem, is chosen. Sterilization. The explant is disinfected to prevent contamination. Culture in nutrient medium. It is placed in an agar-based medium with essential nutrients and growth hormones. Callus formation. The tissue forms a mass of undifferentiated cells. Shoot and root development. Growth hormones induce shoot and root formation, hardening and acclimatization. The plantlets are transferred to the soil to adapt to natural conditions. Advantages: rapid multiplication of plants, disease-free and genetically identical plants, conservation of rare and endangered species, used in agriculture, horticulture, and forestry. Examples: bananas, orchids, sugarcane, potatoes, and medicinal plants. Humans have turned to artificial forms of vegetative propagation for several reasons. Faster plant reproduction. Artificial vegetative propagation, such as grafting, cuttings, layering, and tissue culture, allows for quicker reproduction of plants compared to traditional seed propagation. This is especially valuable for commercial purposes where rapid production is necessary. Preservation of desirable traits. It helps maintain specific desirable traits in plants, such as fruit quality, flower color, or disease resistance. When plants are grown from seeds, genetic variation can lead to offspring that may not have the same favorable traits as the parent. Production of plants that do not produce. Viable seeds. Some plants either do not produce seeds, produce very few seeds, or the seeds are difficult to germinate. In such cases, vegetative propagation is the best way to reproduce these plants. Improved yield and quality. Techniques like grafting can combine the desirable traits of two different plants. For example, a disease-resistant rootstock can be grafted with a high-yielding fruit variety, resulting in a plant that has both qualities. Uniformity in plant production. It ensures uniform growth and appearance of crops. This uniformity is especially important in agriculture and horticulture, where consistency in size, shape, and quality is crucial for market standards. Bypassing long juvenile phases. Some plants have long juvenile phases before they can produce flowers or fruits. Vegetative propagation can help bypass this period, enabling plants to mature faster and start producing earlier. Restoration of damaged plants. It is used in the repair of damaged plants or parts of plants. For instance, grafting can be used to replace damaged branches or parts of a tree. Thank you for using your time to learn. Subscribe, like, and share. Gain access to our well-illustrated notes, activities of integration, assessments and quizzes, and exciting project work.